Good morning to everybody who is participating in this talk. I would like to welcome Dr. Marc Hellmuth and thank him for having accepted our invitation. Dr. Marc is an associate professor at the Department of Mathematics at the Stockholm University. He had his PhD in computer science from the University of Leipzig, and he has ongoing collaboration at many international universities like in United States, Austria, Denmark, Canada, China, and Slovenia. His projects are interdisciplinary, including topics in mathematics, computer science, and bioinformatics. The focus of his research is on the mathematical characterization of data and structures and the design of efficient algorithms besides the theory of complexity. In particular, he works on the development of mathematical theory and algorithms to help understanding complex life sciences processes. The title of the talk of Dr. Marc today is Gene Family Histories and Homology Relations, as you have seen in the presentation first. Dr. Marc, and I have some advices here. Dr. Marc will talk to us first, and after, the participants are free to make questions to our speaker. You can write the questions in the channel, I can see them, and I will read them from Marc. If you can formulate the questions in Portuguese, we can translate them. Now, I will speak, Marc, I will speak a bit in Portuguese to our participants to let them know this. Então, bom dia, pessoal. Se vocês vão ouvir a palestra do Marc, e aí, se vocês quiserem fazer perguntas em português, porque muita gente entende a fala, mas tem mais dificuldade de falar, né? De perguntar em inglês. Então, vocês podem perguntar em português, tá? E nós, a Raquel e eu, vamos traduzir aqui. Então, fiquem à vontade, tá? So, please, Marc, it's with you. Thank you again. Marc, I think you are muted.
Hello. Oh. Thank you. Yeah, thank you for the hint. Hello, bon dia to Brazil, and good afternoon from Stockholm. And it's a pleasure to be here and to talk here. And thank you very much for the invitation. So in particular, thanks, a lot of thanks to the organizers and for the talks that we already heard. So today I'd like to talk about gene family histories and homology relations. And to this end, I will start with a short introduction. So I need to go here. Okay, so let us assume that we would know the true evolutionary history of these species here. And this history is usually depicted as an evolutionary tree. That is, we have a rooted tree with ancestor species here on top. And we simply will consider the species in this talk as labeled with capital letters here. When we talk about species, we usually think about their genomes, right? So a species is essentially a set of genes, so to say, and each of these genes also evolve in a tree-like fashion along the species tree here. You can see here on top a single ancestral gene, and this gene evolves here along this blue species tree. And you can see that there are several colors on the gene tree, and they reflect different mechanisms that can act on the genes through evolution. So we have here red bullets, which are so-called speciation events. We have here these blue squares, which are duplications events, and we also have here such arcs that are rooted at such a green triangle, and these are horizontal gene transfer edges, and these are the mechanisms that essentially act on the genes through evolution. So speciations are the point where the species diverge. Duplications are simply the duplicates. We get a duplicated gene within a lineage, and we have here also some sort of losses. A gene may get lost through evolution, and we have here also things like horizontal gene transfer, the transmission of genes across distinct species. If I now give you the true evolutionary history, what would this mean? It means that you have full information of all the relationships between the genes. So, and this is what we call homology relations. Homology relations are binary relations between genes. And there are several types of such homology relations. We have orthology relations, paralogy, best matches that are defined in terms of so-called evolutionary closest relatives, or xenology, which is defined in terms of horizontal gene transfer edges. And the point is, the knowledge of these homology relations is extremely important. Yeah. So, without the knowledge of these relations, biologists could not continue in many cases with their work. Yeah, they are important to understand the function of genes, for genome annotation, and therefore are important implicitly for medicine, personalized medicine, or drug development. They are important to understand mechanisms that act on the genes through evolution, or also to construct certain trees. Okay. In summary, plenty of homology relations exists. There is only a small little detail here. They are defined in terms of the true evolutionary history. So, the point is, we don't know this history. Yeah, and we will never know this history with certainty, because we cannot observe the past. The only thing that is essentially available to us is the genes from these species that live at the bottom of this tree here. So to say, the currently living species and their genomic material. So, and we only have the information of this green box here available, and that's it, right? So what now? How can we get these homology relations that seem to be quite important? Okay, so one way to do this problem of inferring homology relations and to attack this problem is as follows. You start with sequence alignment. You collect the genetic material of the species you are interested in, or the relations between the genes you are interested in. And based on these sequence similarities, sequence alignments, you can get sequence similarities. And based on this, you can use some sort of tree-free magic. And we will treat this here as a black box. Yeah, if you have questions to that, I can answer questions to this after the talk. But there is some tree-free magic that makes no assumption about the trees on top. Sorry. And then we get an estimate of such homology relations. And since these estimates are, or these homology relations are binary relations, we can depict them as such a graph, where the vertices are the genes and edges between tell us which of these genes are in that relation and which of these genes are not. So the crucial question is now, when are such estimates reliable? Yeah, I use some fancy tools, tree-free magic. I get some estimate and now I want to say something about that data. Can I trust in that estimate, right? So, and I will explain in some more detail on how one can attack this question on two specific homology relations, the orthology relation and best match relations. What is orthology? So two genes X and Y are orthologs if they were separated by a speciation event within the true history. So let's have a look at this evolutionary scenario. We have here a species tree with an embedded gene tree, and whenever the vertices of the gene tree reside within the vertices of the species tree, that is, they are within these ovals, we simply color them red. So red vertices specify speciation events in our gene tree. So if we have this coloring, then we can ignore the species tree because we already know where the speciations are. We can also ignore all the other colors of this tree. So essentially we end up with a red-black labeled tree where we have the speciation events colored. And now, when are two genes orthologs? When they are separated by a speciation event. That means, in a mathematical way, that if we have such a colored tree and the last common ancestor of two genes is a red vertex, then it is a speciation event, and this means, and this is the definition of, these two genes are then orthologs. And in this way, we can infer from that tree this particular orthology relation. Yeah, depicted as a graph. By way of example, A1 and B1, their last common ancestor is here, this red vertex. So they were separated by a speciation event, so they are orthologs. And if we have a look here, A1 and B1 are in fact connected by an edge, right? Or if we have a look, say, to C1 and B2, they are not. There is no edge, and if you look at our tree here, C1 and B2, whereas B2 is here, and if we go here and here, we see that the last common ancestor is this vertex. It's not a red vertex, and so, yeah, well, they are not orthologs, and correctly, there is no edge here. So, the orthology relation comprises all pairs of such orthologous genes, and what you can see in this graph already, that this is a binary relation that is symmetric, but not a transitive relation. Okay, so back to our observation. We have these sequence alignments, and then we use some tree-free magic, and there are plenty of tools that have been established, like protein ortho, InParanoid, OMA, eggnog, and so on and so on. And all these tools allow us to infer a binary relation, such a graph that is then an estimate of the orthology relation. And back to our question, how can we trust such estimates? I mean, we don't know the truth. And at least to attack this problem to some extent, and we provide here a necessary condition for such an estimate to be biologically feasible. And this is what I call a valid estimate. And an estimate of an orthology relation is valid if there is a tree together with a red-black labeling such that the last common ancestor of two genes X and Y is colored red, if and only if this pair was estimated as orthologs. So in other words, an estimate is valid whenever I can find a tree that supports my data. Yeah, if I can not find any tree at all that supports my data, I mean, then for sure I must have done some mistake. Yeah, by way of example, I give you this estimate here, and now I ask you, is this a valid orthology relation? I mean, is there a tree at all that supports my data? And the answer is yes, right? We have already seen the tree from which we have derived this relation here. But yeah, I mean, this is the easy way. We have already seen we got a tree, but now I give you an arbitrary estimate and ask you still, is there a tree? So can we mathematically characterize valid estimates of such orthology relations? Good. I mean, from the simple point of view, what could we do? We could take such estimates, or say valid orthology relations that we have obtained from, say, some simulations. Yeah, we have simulated gene trees. We can very clearly say which of these pairs are orthologs and which not, based on these simulations. And then we could look at the valid orthology relation, which may look like this graph here. And then you can play around a bit with these structures and try to figure out if there are certain substructures that are overrepresented or underrepresented, and just to get some idea of such graphs. And if you do this long enough for valid orthology relations, then you will observe that there is one substructure that never occurs. And these are induced P4s. When you have a valid orthology relation and you take 4 vertices into account and all edges in between them, and this thing looks like a path, yeah, then this is called an induced P4. But such substructures will never ever occur in valid orthology relations. Yeah, you can have on 4 vertices such a substructure is possible, or a structure, an induced subgraph that looks like this one, or like these ones, but never induced P4s. And this, of course, leads to the conjecture, I mean, if I cannot observe this in any of my valid orthology relations, I mean, is it true that this does this give us a characterization, right? And so the non-existence of induced P4s is furthermore supported when you look at all possible representative trees on 4 leaves. Yeah, I mean, these are not all, but all representative trees on 4 leaves. So if you have, representative means that that there are further trees, but all of them would, in some or the other way, explain the same subgraphs that are shown here. Good. So let's have a look to this tree. This tree here encodes an empty relation. None of them are in that relation. Or we have, say, this tree here. Then it is possible that all of the genes are pairwise in this orthology relation. Or we only have one pair, maybe. Yeah. So, and if you look long enough on these 4 vertex-induced subgraphs, then you can see that these are all subgraphs on 4 vertices, all possible subgraphs on 4 vertices except for one, and this is again an induced P4. So in this slide should give you somehow the idea of why it is not possible to encode induced P4s by such gene trees that have, say, speciation and other events. And in fact, this is now the characterization that we came up in 2013, and this is an estimated orthology relation is valid if and only if its graph representation does not contain induced P4s. And for those that are familiar with graph theory, immediately there rings a bell, because the class of graphs that do not contain induced P4s have been excessively studied since the 70s already. So this is a very well understood graph class, and it's called complement reducible graphs, co-graphs. And there is then a further characterization in terms of symbolic ultrametrics that is essentially a generalization of a metric, not to numbers, but to combinatorial objects, so to say. And this is a work of Böcker and Dress, where we heavily reused many of their results in order to come to our result that is shown here. A nice thing is now, if I have a co-graph, or a graph that does not contain induced p-force, in which case I have a valid estimated orthology relation, I essentially get for free a tree, and this goes as follows, and this is based essentially on the idea of these complement reducible graphs, which I do not define here, but I will explain shortly how we get the gene trees now for free. In our case, the graph is disconnected. So what we do is we start with a root that is labeled black. And then we attach the connected components to that vertex. In our case, we have a single vertex component A and a component that consists of the vertices B, C, D, and E. What we do now is we go into those connected components that are not single vertex graphs and build the complement. That is, we go to this connected component, we remove all edges that are in there and add all edges that are missing to get the complement. So the complement looks like that. And now we attach these connected components to that vertex. Now we go and recurse on the connected components. We go to the connected components, build their complement, and attach them. And then we I'll label these inner vertices here with black, red, black, and so on. And this way, we get for free the tree that represents our estimated orthology relation. And this is essentially why this always works, is based on the idea of complement reducible graphs. So, based on all of these results, and in particular also the results for complement reducible graphs, it is possible to recognize valid orthology relations in linear time. In other words, one can say in linear time whether a given estimate has a supporting tree or not. And this can be done in linear time. And then the gene tree can also be found in linear time. And we have a nice forbidden subgraph characterization, and this can be used to show that the underlying correction process, if you have a fixed parameter tractable. As a byresult of all of these mathematical studies that we had, we got a very nice paper. And the content of this paper is essentially that we used these tree-free estimates, and in this case we used protein ortho, to get some estimate of an orthology relation, and then we designed an integer linear program to optimally correct these estimates, and to get also a full evolutionary scenario that explains our relation here. So we have chosen an exact method. So integer linear program can be used to exactly solve the problem. So in particular, because we wanted to have a proof of concept. And what we have observed is essentially that for genome-wide data sets, so we have many gene families, the corrected orthology relations can be used to infer not only the underlying gene trees, if I had shown a few slides before, but also the species trees and their reconciliation. So all that information is entirely contained in these corrected graphs here. And so, this software is implemented. It's called Paraphilo, and you can use it if you want. And a second observation is that we had was that noise in the data, so to say, hand-waving noise in the data, can be used as an additional source of information. What I mean here is that the paralogs, which are defined in terms of gene duplications, that are those pairs for which the last common ancestor is not a red bullet, but say a blue square. And in many studies on genomic material, these paralogs are treated as very dangerous noise that has to be detected and removed, which reduces your data sets essentially to so-called one-to-one orthologs. And we have shown that this restriction is not necessary, and that in fact the paralogs are the key elements to find full evolutionary scenarios. So if we would not have, so to say, enough paralogs, we would not be able to have enough information about the species tree. So as I said, this was just a by-result based on the motivation to understand the mathematical structure of orthologs. And what I want to emphasize with the sentence is that it is quite important just to play around from a mathematical view on problems and try to understand them. And sometimes you get some nice solutions for free. Okay, so now we know that valid estimates are not allowed to contain induced P4s. They must be P4 free. They must be a co-graph. And there is a small little problem here that the editing process, that is the addition and deletion of edges in our orthology graph, such that I get a co-graph, this is an NP-hard problem. And Paraphilo that we have established uses interlinear programming to compute exact solutions. So, I mean, this is, of course, then only feasible to a certain extent. So we had done some simulations, and we also had real data sets. And so for 10 species, the species tree has 10 leaves and 1,000 gene families. That is, we have 1,000 gene trees embedded into this species tree. It costs around 2 minutes to get an exact solution for a valid estimate plus the full evolutionary scenario. For 18 species and around 8,300 gene families, it takes already 45 minutes. So, I mean, it's doable, right? But yeah, we want to have it faster, of course. So more efficient methods are needed, and the bottleneck in particular is the co-graph editing problem here. And what you can see, if I give you this induced P4, and I'll ask you, how many ways are there to edit this P4, right? So you could, for example, remove this edge to get a graph that is not a P4, or you can remove the middle edge, or you can add edges, you can remove add edges, and so on. And so there are plenty of ways to edit a given induced P4. And this leads to the question if there is an idea, if you could guide the process of editing P4s in some meaningful way, right? We have biological data, and so is there some biological reasoning behind to extend, to delete or add certain edges? And this leads to the concept of best matches. So best matches are a second homology relation that I'd like to introduce here. And for the best matches, it is the inner colors that we had before, these red bullets and blue squares, are not so important. For the best match, it is important to know from which species the genes come from. And therefore, we consider them here as leaf-colored trees, and the colors of the genes simply reflect from which species the genes are. So we have in our example a red species, a blue species, and a green species. And since we are not really interested in this coloring here because we don't know it at all, right? So we simply omit it here and just look at this gene tree here. And now this is a definition, and let's take some time to understand this. This is not so easy. So a gene Y from a species Y is a best match of a gene X and taken from a species X, and then we write X points to Y if first two species are different and the last common ancestor of X and Y is below or equal to the last common ancestor of X and Y prime for all the genes from species Y. Let's do that on an example. So we have here all these genes, the leaves of this gene tree, and based on this gene tree we can infer the best matches. Okay, let's start with A1. What are the best matches of A1? And now we fix a species because we restrict best matches only between two species. Say the best matches of A1 from the blue species. Okay, so we first compare A1 and B1 and see their last common ancestor is here, where the circle is. And then we have A1 and B2. And there you can see that their last common ancestor is located here. So in fact, the last common ancestor of A and B1 is below the last common ancestor of A1 and all the genes in color blue. Yeah, so it's precisely that point, and this is why A1 points to B1. That is, B1 is a best match of A1, and B2 is not a best match. We could have reciprocal best matches, like in this case, that is, A1 is also a best match of B1. Why is this the case? Yeah, if you look at, if you want to now the best matches of B1 and with respect to the red species, then we compare B1 with A1, here the last common ancestor, B1 with A2, the last common ancestor here, and so again we see that A1 is now also a best match of B1, and in this case we have a bidirectional arc, and which gives us reciprocal best matches. And now we can play the game for all the other genes as well, and at the end of the day we get a best match graph that reflects this relation, which is not symmetric anymore, and yeah, here all the best matches are listed. The nice thing again is we can use sequence similarities in order to infer, say, maybe with BLAST or other tools, best hits, and we can then translate them into such graphs here. And again, the question is, how can I trust now this estimate? And we can say they are valid if I can find at least a tree that supports my data, right? If I cannot find a tree at all, then I must have done some error somewhere, some mistake somewhere. I just sketch here that the characterization of these best matches is quite involved. So I will omit this characterization here. The main idea is to transform the graph structure of these best matches into, say, further combinatorial objects, and for example, so-called triples, small little trees, and then you combine them in a certain way. But you simply transform the structure into further combinatorial objects and use then the structure of these combinatorial objects in order to see whether these BMGs are valid or not. The important point is that the recognition of valid BMGs and also their tree reconstruction can be done in polynomial time. So this is not a difficult problem. And what I'd also like to say is that these estimated best matches are the basis for many of the commonly used methods for orthology inference. Yeah. So at the heart of these orthology inference tools, these tree-free magic tools, there are best matches. And the question we had is therefore, can we use now the mathematical structure of these best matches to improve orthology inference, so which means to delete the correct edges from possible occurring induced P4s. Okay, so here is a full evolutionary scenario, say a true one or whatever, or simulated one, and based on that you can infer this best match graph. Okay, now if we look at the orthologs, those ones for which there is a, it's not red anymore, but still it's a bullet. Whenever the last common ancestor is a bullet, there are orthologs. By way of example, D1 is orthologs to all of these genes, and you can see here already that D1 has a reciprocal best match, right, to all of these. So I color here all the edges of our best match graph that also yield, or that are also orthologs. And the observation is that there are overlaps between the orthology graph and the best match graph. And what we have shown is that in duplication loss scenarios, that is, in the absence of horizontal gene transfer, all orthologs are also reciprocal best matches. And this in particular implies that the orthology graph is always a subgraph of the best match graph. And this is interesting because if you have false orthology assignments in there, and you look at your reciprocal best matches as, say, as guide, then there are only false positives essentially in there. So the editing process reduces to a deletion problem to destroy P4s, which is still an NP-hard problem. But anyways, what we want to understand is the dependencies between such best match edges and the orthologs. Okay. Which of the reciprocal best matches are orthologs or not? This is the central question. And you can see here an example where, I have drawn here an evolutionary scenario, and the best matches are X and Y. So they are both best matches of each other, that is, they form reciprocal best matches. And this is simply due to the reason because there are only two genes, right? And if you look now at the orthology relation, you can see, it's not a bullet, the last common ancestor of X and Y, so they are not orthologs. But on the other hand, I can find a further example that gives you the same best match graph, but the last common ancestor is now a bullet, and so they are orthologs. So on a first glance, it is not reasonable to think that this is easy to infer these directly when you only look at two species. And so the idea is not to look only at the best matches between two genes of two species, but take a third species into account, say the green one here. And now it becomes interesting. What one can prove is that if you find such an induced subgraph in your best match graph, then there is only one scenario, one possible scenario that can explain this substructure. And the only scenario that exists to explain this scenario or this best match graph here looks like that. So in particular, the last common ancestor of X and Y must be a duplication. So for every scenario that explains this best match graph, it can be mathematically proven that their last common ancestor must be a duplication. And this means that this reciprocal best match here cannot be an ortholog. We must remove it, right? If you take this graph, then if you look at the orthology relation, the estimated, these would be the bidirectional arcs. This would lead to an P4, but we know at that point that we need to remove this edge here, which leads to this orthology graph on the right side. So, and what we wanted to understand, if there is a way to unambiguously detect, to detect all unambiguous false orthology assignments. That is, we wanted to detect all false orthology assignments for which there is no evolutionary scenario at all for which these pairs of genes could be orthologs. And in this case, they are false orthology assignments. And there are many, many motifs in such BMGs that lead to unambiguous false orthology assignments. These are the red arcs depicted here. So if you find this graph, we have seen it in the previous example, then this edge must be removed, for sure. If you find such an induced subgraph, you can say with 100%, at least in these DL scenarios, that this and this edge must be wrong. And so on and so on. And this is a bit more complicated, the hourglass chain. And at a first glance, it looks like, if I want to find these false orthology assignments, I must detect these subgraphs. And this is here some arbitrary large subgraph. And subgraph isomorphism is not an easy problem. The good news are we don't need to find these motifs explicitly. This is not explained here, but you use instead a so-called augmenting tree to stepwisely look where these false orthology assignments are located and to detect them. Essentially, these substructures here are just a vehicle to prove what's going on. So, and the theorem that we had is that all unambiguous false orthology assignment can be detected in polynomial time. And the resulting corrected estimated orthology relation, after removal of all these unambiguous false orthology assignments, is P4 free, that is, it is a co-graph, and therefore it must be valid. And yeah, and then we have simulated some evolutionary scenarios, and this is shown in these plots here. What can you see? You see first, each point in this plot here is a single evolutionary scenario. Yeah? And on the x-axis, there is the number of duplication events, and on the y-axis, there is the number of loss events here. So to say, as more it goes in this direction, as more complicated the scenarios are. And the colors here indicate the number of faults, or the rate of faults orthologs. Yeah? What does it mean? We have an evolutionary scenario, and now we can correctly infer all the best matches and also all the reciprocal best matches. And now assuming that all reciprocal best matches are also orthologs leads to faults orthology assignments. And then we looked at all the unambiguous faults orthology assignments in these graphs, in these estimates, and removed them, and then we obtained this graph here. So as darker the color is, more faults orthology assignments we had, and if the color is yellow, then you can see that we had nearly perfect orthology assignments. And you can see that the removal of all unambiguous faults orthology assignment leads to a perfect orthology assignment in most of the cases. There are some exceptions, and these remaining faults orthology assignments are, I would say, are not identifiable in the framework of best matches and are quite often associated with a large number of losses for a given number of duplications. So, but this is promising, this result. Good. To summarize at that point, homology relations are binary relations between genes. And the understanding of these homology relations is not only great fun from the mathematical point of view, but provides also deep biological insights and also improvements for their inference. And I explained it in some more detail on the orthology and the best match relation. But there are plenty more of them. But in general, I mean, this is a quite generic approach here that is applicable for any data that is inferred from some source of information, right? You just first need to analyze the data, find possible patterns, use these patterns to characterize true or correct or valid, feasible, however you'd like to call it, data, and then use these characterizations to correct your estimates and to prove or find new inference methods to get reliable estimates. And what are our next steps is, I mean, as I said, usually these best matches are used to infer these orthology relations with certain clustering approaches and so on. What we aim currently is to replace this by the best match graphs and to extend established pipelines, in particular protein ortho, to find reliable orthology assignments. And many of these details I gave here work very well in the absence of horizontal gene transfer. And what's one of the main steps we are currently working on is methods to automatically detect horizontal gene transfer. And that's it. I'd like to thank, of course, my co-authors and colleagues. So in particular, Manuela Geis and David Schaller, who both work on these topics on best matches and orthology inference and heavily in their PhD thesis. So both have finished their PhD. And of course, Peter Stadler, one of my main collaborators, and Maribel from Mexico, Vincent, Casey, Marcus, Martin, Nicolas, all of them have contributed in one or the other way to the topics I presented here. And thank you very much for your attention. And if there are questions, I'm here. And just a short announcement, there is an open postdoc position in my group in Stockholm. So if you are interested, just click on that link and look for biomathematics postdoc and then simply apply. Thank you.
Thank you, Marc. Thank you, Marc, for your nice presentation. And we have some questions here. I don't know if you can see directly in the comments.
Let me.
Or
Yeah.
I can read it. Okay.
Yeah. If you can read it.
Yeah. Yeah. Some people like it's on the screen. Do you see? Some people like to call out paralogs for when 20 genes form two groups of 10 because there is no example of cohortology. Do you split graphs in that case? And often out paralogs have distinct gene names, I guess.
No, we distinguish here not. So, I mean, orthologs are not distinguished, so we do not subdivide them into the group of in and out paralogs. This is one mix, so to say.
Okay. I have one question. It's not properly a question, but you are talking about orthologous genes, right? And we have some difficulties to distinguish between orthologs and paralogs. And so maybe you talk a bit about this distinction because it introduces difficulties in the algorithms, right?
Yes. So I know that there are, for some reasons, some definitions of orthologs in terms of function. So two genes are orthologs if they have the same function or something similar. And I just want to clarify that this is not what I talk about. So
Yeah.
it's really the last common ancestor was a speciation event, or it was simply not a speciation event. And this gives a clear distinction between orthologs and non-orthologs.
Yeah.
And from the mathematical point, there is nothing to discuss there or to yeah. And yeah, does this answer your question or?
Yeah. No, it's just to distinguish this because there are other names. One of our participants is telling we have these in paralogs and out paralogs and things like this, right?
Yeah, but they are all under the common framework of orthologs, and the in parallel, these are under the common name of paralogs. So, and then you can, of course, start to distinguish them also a bit more, but this is not what we have done in our work.
Yeah. We have another question here. Raquel, can you put this in the screen so Marc can read it? This BMG runs in parallel. Is there a limit of species that can be used with this algorithm? For example, increasing the number of species will be time consuming?
Yeah. Okay. I need to check the real runtime, and there is a reason why I wrote polynomial time. I don't have the exact runtime currently in mind, but it's not linear. So there is a factor of, so it's a like quadratic in the number of species, but I'm not sure. I need to check. So first point is that we have these algorithms implemented. They are applicable. They are also working for a large number of species, I guess. But I cannot tell you precisely if this is hundreds of species or 50 or things like that. Oh, now I don't have the question anymore. And what was... All right. And can it run in parallel? This is a very interesting question. And yeah, I don't see that this is a problem. But it's a good question, but I need to think about that in more detail.
Okay, and another thing you mentioned in your presentation is to tell a bit about the black panel and the tree-free magic that you mentioned.
Yeah. Then I need to share my slides possibly. Yes, thank you. So, okay, so good. So this here is a perfect example. And so what you can see here is again a gene and a species tree, and you can think about the time axis here as so to say divergence time of two species. And so in this perfect example, we would say it's okay. Y is an orthologous to X if the species are different, and their divergence time, the divergence time of these two genes X and Y is, so to say, equal to the divergence time of the underlying species, right? So the divergence time of the species A and B, which is this time point, so to say, is the same as of these two genes here. So, and for example, B1 and B2 and A2, the divergence time in the gene tree is along these edges here. So it's not the same as the divergence time of the species in which they reside. But I mean, this is just hand waving the very rough idea. And so instead of looking at divergence times, which we don't know, one can look at sequence similarities. So that is that, yeah, we take the closest similar genes. So as close as the genes they are related as, or that, yeah, as smaller their divergence time is, as closer they are related, as more similarity do they have. So, right, and this leads then to the concept of these best matches, which is expressed by these sequence similarities, and then we look, of course, to the reciprocal ones. And this cannot work perfectly due to several examples, and this example was shown already in one of the slides. But it does not, at least it does not assume that we have a constant molecular clock. This is something I'd like to emphasize. It just need to be ensured that within the evolution of one gene family, the evolution rates are not too weird. That's essentially the only thing that is needed. So what you do is you take sequence similarities, and the motivation why you do that is behind the assumption that, yeah, the time the species diverged is approximately the same as the time the genes diverged, which is approximately expressed somehow in sequence similarities. So this is the main idea.
Okay.
So, and then you don't need these trees anymore. So you only look at the gene sequences and their similarities.
Good. I'm not. Okay. There are some other questions here. No. And I think that I was curious, and you didn't went in your talk. But could you talk a bit about horizontal gene transfer? Because this is another question that is very difficult to deal with in algorithms, right?
Right. The first thing is what we have shown. I mean, if you again, if you have a valid that comes from some gene and some species tree. Then one can show again that all the information about the evolutionary scenarios is contained in these homology relations. There is one small little problem, and this is you are not interested in a gene tree with a particular labeling anymore. I mean, this is a necessary condition, but you must also ensure that there is a species tree such that you can embed your gene tree into the species tree in a time-consistent way. And this makes the most headache. So this is one problem, but given a valid relation, all these things can be shown. When does it work? When does it not work? Now, the question is, of course, how to get estimates of such relations. And one way to address this problem is instead of directly inferring horizontal gene transfer, so saying between which genes there was horizontal gene transfer or not, try to infer groups of genes for which you can ensure that no horizontal gene transfer occurred. Then you have groups of genes where you know there was no horizontal gene transfer, and then you start to filter out which of these genes were in the putative true history a possible horizontal gene transfer event happened in the gene tree, and that gives you then information about these genes that were separated by horizontal gene transfer. And this is where we are currently working in, and it looks promising so far. So it keeps us moving. Yeah.
Okay. I guess there are no other questions. So I would like to thank you, Marc, again for being here with us today. And we learned a lot. And
thank you very much.
I hope to see you in person in Brazil sometime.
Yeah, I hope so too.
Yeah. And so we can finish this session. And remembering that the talk of Dr. Angelica will be today at 2 p.m. or at 14, okay? So I think, Raquel, we can finish this, right? Thank you.
Bye-bye. Ciao,
Thank
ciao.
you all. Ciao.